evening, everybody. I'm Jinliang Wang, as said by Louis, from the Institute of Zoology, Zoological Society of London. Here I'm sitting in the morning of London. And hope everybody has enjoyed Koloa these days, the beach, the forest, and most importantly, the excellent talks. I certainly enjoyed listening to a couple of these talks. I learned a lot in the past few days. Anyway, today we have session 11. It has 3 parts. I will be presenting the first part, mainly about the concept, the theory, and the methodology of estimating relatedness using molecular markers. And the second and the third parts are going by Louis and Natasha. They will present more interesting stuff about using the R code generated by Louis to analyze some empirical data sets. I first give an overview of what I'm talking about in the next 15 minutes, something like that. So what I'm talking about is genetic relatedness. It's not, other relatedness like, social relatedness or geographic relatedness. It is genetic relatedness. And why we are interested in estimating relatedness. And then I give some brief discussion of the properties of genetic relatedness as estimated from genetic marker data. And briefly show you the methods available for estimating relatedness. So these include pedigree-based methods and marker-based methods. Of course, I will focus on the second part, that is marker-based methods. Now, I briefly discuss with you some of the issues in related estimates from marker data. And hopefully, at the end of the 15 minutes, we get some conclusions. So first, what is relatedness? What I'm talking about. So from the genetical, genealogical point of view, genetic relatedness measures the degree that 2 individuals are genetically related due to recent common ancestors. So here, the most important thing is common ancestors. The extent of relatedness is determined by the number of common ancestors and the number of generations linking the 2 individuals to each common ancestor. So the closer the common ancestors are to the 2 individuals, the more relatedness. And also, the more common ancestors the 2 individuals have, the more related they are. Here, implicitly, we have a reference population where it is assumed there is no inbreeding and no relatedness. So relatedness is measured relative to this reference population. If you change the reference population, you will change the relatedness significance, its values, and its interpretations. We will come back many times to this issue later on. From the genomic point of view, genetic relatedness is measured by the proportion of genes shared between individuals that are IBD, where IBD genes are copies of the same gene of a common ancestor. So genes IBD are different from genes IIS, that is identical in state. When genes are IBD, they must be identical by descent, but the other way around is not necessarily true. Sorry, I think there is a reminder here. I did dismiss this. Okay. So again, there is a reference, implicit reference population here in our definition, where the reference in the reference population, we implicitly assume that there are no genes that are LBD within and between individuals, and relatedness is relative to this reference population. So this is equivalent to the genealogical definition. So relatedness is really a probability in the continuous range between 0 and 1. 0 means no relatedness, no related, while 1 is completely related. So here are some common relationships. For example, the first degree relatedness refers to parent offspring and forceps. Their relatedness is 0.5, while the second degree relatedness usually refers to half-siblings, avicular, grandparent, grand offspring, double first cousins. Relatedness value is 0.25, and so on and so forth. Commonly, relatedness is also measured by coancestry coefficient or kinship coefficient. It is defined as the probability that two homologous genes, when taken at random from each individual, are LBD. For example, for forceps, we have coancestry coefficient 0.25, while its relatedness is 0.5. For half-siblings, it's half the values of forceps. So from this, it seems that there is a fixed relationship between relatedness and coancestry. Relatedness is always 2 times the coancestry coefficient, but this is not true. This is just for non-inbred individuals. The general relationship is given by this little formula. So only one fx, fy equals 0, we have r equals 2 theta. Otherwise, r is smaller than 2 theta. Let's see a few examples. For example, for forceps, whose parents are full siblings. So individual x and y are inbred. They have an inbred coefficient of 1/4. In this case, if you use the formula to get the exact R value, it is 0.6. But if you assume R is 2 times theta, it is 0.75. It is an overestimate, obviously. And the overestimation by that simple relationship is most extreme with completely inbred twins. If you use the formula, you get relatedness between completely inbred twins, a value of 1, which is correct. But if you assume 2 times theta, it is 2, obviously too high. So why we are interested in estimating relatedness? There are many applications in various fields. For example, in our human society, marriage and inheritance laws are partially determined by R. And in forensics, for example, relatedness is used for identifying bodies and body parts. And nowadays, with the use of internet and large SNP data sets, it's very common for a few companies to reconstruct pedigrees using genetic marker data, so you can find your remote relatives in some other parts of the world easily. And in QTL mapping, relatedness is used for finding disease genes, and in domestic animal and crop breeding, we can use relatedness to estimate heritability of a quantitative trait, so that we can improve the trait by selection. And in conservation genetics, there are many more applications. For example, we can select individuals, unrelated individuals for reintroduction and for selecting unrelated individuals to reproduce, to maximize genetic diversity in the next generation. And in evolutionary biology, we could use inbreeding and relatedness to make some tests or some hypotheses like kin selection, inbreeding depression, mate choice, all those kinds of important concepts can be tested with the use of relatedness. And there are many other applications. So the properties of relatedness. In the definition, from the probability point of view, it's a probability. So it is a value in between 0 and 1. But there is another definition, which is correlation-based. It's a correlation coefficient. So its legitimate range should lie between -1 and 1. -1 means they are completely unrelated. They are much less related than the average of the population. So it's again, it's a relative quantity relative to the reference population. But the reference population here, in both theory and in applications, is quite weak, as I will show you later on. Or I would say it is arbitrary. Anyway, at least in theory, in the reference population, we assume genes are non-IBD. And so all individuals are outbred, inbreeding coefficient equals 0, and unrelated. That is, relatedness equals 0. Of course, the reference population is artificial. It is arbitrary, but it is important in determining R, as I will show you in the example later on. So here is the example. It's a 4-generation pedigree showing the relationships between individuals. Well, here we focus on the 7 individuals in the 4th generation at the bottom. So when we have no pedigree records at all, that is, we have no pedigree, then we have to assume most often these individuals are unrelated, because that is more often the case if we have no records of whatsoever about the individuals. So we assume they are unrelated. Their relatedness is 0. Now, after collecting some data, we get a 2-generation pedigree. And from this 2-generation pedigree, we can see there are actually 2 full sibling families. The first full sibling family has 3 individuals, and the second family has 4 individuals. So their relatedness within the family is 0.5. While between families, we have no information at all. So they are unrelated. Relatedness between families is 0. Now, we collected more information of their ancestors. So we get a 4-generation pedigree. So in this more complete pedigree, we can see all individuals are related. Importantly, the 2 full sibling families, they actually share grandparents. So they are actually related. So what I really mean using this example is that in the pedigree analysis, and it is also true with marker analysis, we always have a reference population. It's more or less arbitrary, but it is important. Remember, this point is important in understanding our estimates. and interpreting our estimates. Yes, so in certain conditions, relatedness can be meaningfully negative. And this is true, for example, when we have some hybrids or immigrants from other populations, and we estimate the relatedness relative to the resident population as a reference. So those individuals, they tend to be less related to the resident individuals. So they have a negative value. And so in this case, it's better to use the correlation definition of relatedness rather than the probability definition, because negative values are impossible with probability. So another point I want to discuss is that, with pedigree-based analysis, pedigree give us an expected value of relatedness. For example, full siblings, they are 0.5. This is true, regardless of the population you are talking about or the species you are talking about. Two full sibling elephants, two full sibling ants, they have the same relatedness, 0.5. This is always true if you are using pedigree, because this is the expected value. It has nothing to do with the genome structure, genome size, and has nothing to do with the species. It's purely determined by the pedigree. It's the expected value. But in reality, the realized or actual relatedness varies around this expected value due to Mendelian segregation. For forceps, for example, different forceps pairs may have different realized relatedness in the genome. I will show you later on. So there is a variance depends around the expected value, depending on the relationship we are talking about. It also depends on the genome size and the structure. For humans, we have 22 autosomes, and the average map length between males and females. By the way, males and females have different map lengths. The average value is 35 Morgans. So using this human genome, we can calculate the variance of the realized relatedness between full siblings. It is 0.0015, and the standard deviation is 0.039. Is it true? There is an empirical study conducted by Peter Fisher, and they looked at the relatedness between 4,400 full sibling pairs. And here, this graph shows the distribution of the estimated values among these 4,400 pairs. The average value of the realized genomic relatedness is 0.498. It is very close to the truth, that is 0.5. And the standard deviation is 0.036, also very close to the expected value of theoretical value, that is 0.039. From this graph, we can see there is a wide distribution of the realized genomic relatedness among human full siblings. I have a full sibling brother and a full sibling sister. I love my sister more than my brother. Why? Because I have more relatedness with my sister than my brother. It's possible, my relatedness with my sister is 0.6, while my relatedness with my brother is 0.4. It's quite possible. but I mean, you know, it's not that explanation that explains my feeling. It's just an example. In reality, here we are talking about humans, but if you are talking about your own species, because the length of the genome, the genome size varies, the real variation in realized relatedness can be different from the human example, but there is always a variance there. We need to remember that. So here in this table, we have a summary of some of the relationships in terms of their single locus variance and the dynamic variance of relatedness. Of course, the expected value is invariable among loci. It is always the same value. For single locus, 4 siblings has the highest variance. It is 0.125. When you make an average across all loci in the genome, the variance is tremendously reduced to this value, 0.00147. But again, there is some variance there, and the standard deviation is 0.039, as we mentioned before. And for second degree relationships, we can see their expected value is the same, but their variance vary, especially for grandparent, grand offspring. It has a high variance relative to half siblings and uncle nephew. Here in this graph shows the distribution of the genomic wide relatedness, realized relatedness for different relationships. As we can see, although 4 siblings and half siblings and the grandparent, grand offspring have different expected values, that is 0.5, 0.25, they are widely different, but their distribution overlap. So that is why in reality, it's a bit difficult to distinguish these relationships, even you are using genomic data, because their real values do overlap. So now we talk about marker relatedness. So with marker relatedness are estimated from a set of markers, and they are for this particular set of markers. But if these markers are taken at random from the whole genome, and there are many of these markers, an average across these markers would give us a close estimation of the genomic relatedness. So this brings us to the question that is, both markers and the pedigree give an estimate of the genomic relatedness. So which is better, markers or pedigrees? I did a study some years ago, published in Theoretical Population Biology, doing simulations to compare the accuracy of pedigree-based and marker-based estimation of genomic relatedness. And the conclusion is, when you have few loci, pedigrees give you better estimation. But if you have many markers, pedigree tend to give you, markers tend to give you better estimates. So in this graph, it shows the changes in the correlation between marker-based relatedness with true or actual genomic relatedness, and the correlation between pedigree-based estimates with true genomic relatedness. As we can see, the turning point is roughly 10,000 loci. below 10,000 loci, pedigrees are better, while above 10,000, markers are slightly better, but not much, as we can see. And this phenomenon is true even we have, say, millions of markers. It's only slightly better than pedigree. But the situation is different with the inbreeding coefficient. When you have millions of SNPs, the marker-based inbreeding coefficient can be much better than pedigree-based inbreeding coefficient. So for pedigree, we usually use path analysis to calculate relatedness and the inbreeding coefficient. So it's basically, the calculation is quite simple in principle, at least. You first find the common ancestors and then count the number of generations from the two individuals to the common ancestor and make a summation over all the common ancestors or all the paths, and you get the relatedness between the individuals, that is individual B and individual C. Here the correlation, the relatedness coefficient is 0.125. It's very simple. But remember, it's the expected value as we discussed before. And often, pedigrees are unavailable, especially with wild animals. And even it is available, it is incomplete. And most often, it's erroneous. And sometimes it is too shallow. We have just, say, 2 or 3 generations. And we have assumed that the ancestors or the founders of the pedigree are unrelated. In fact, this is not true, especially with the small populations in the wild. And also, it is very expensive and time-consuming to get a decent pedigree. When I say a decent pedigree, I mean a pedigree with complete, with a decent completeness, and with quite a few number of generations included in the pedigree. So alternatively, we can use genetic markers to make an estimation of relatedness. Relatively, there are quite a few advantages with markers. First, it is fast, obviously. We just need a single sample from the current population. We do not need to accumulate our information over generations, which is time-consuming. And it is also cheap nowadays with next generation sequencing. It's relatively cheap. And also it's more accurate. I put a question mark there because, it really depends on the depth of the pedigree if we have, and also the number of markers we have, the quality, missing data, all those factors would affect the accuracy. But in theory, we can get sufficient marker information to get more accurate estimates of relatedness than pedigrees. And for this purpose, that is a marker-based estimation of relatedness. We have developed many estimators. They all based on the allele frequency estimates in one way or another. Comparing these estimators, they all based or rely on the statistical framework linking the observed genotypes, that is, the alleles in IIS, identical in state, to the probabilities of IBD and the frequencies of alleles in the population. I'll give you a very simple example to illustrate this point. Let's consider the inbreeding coefficient it only involves one individual, and we consider a single locus with two alleles, big A and small a. For this individual, Z, we have its parents, X and Y. Obviously, the inbreed coefficient of Z is equivalent to the coancestry between its parents, X and Y. If we know the allele frequencies of these big A and small a alleles, and also we know the inbreeding coefficient, Fz, we can write down the probability of each of the three possible genotypes, big A homozygote, small a homozygote, and the heterozygote, and show in these three equations. And these three equations can be used in two purposes. One is for prediction. That is, we can use these three equations to predict the occurrence of the three possible genotypes, given the IBD coefficients, that is, the inbreed coefficient of individual Z, and the allele frequencies. It can also, we can also turn this around and use the same equations to make an estimation of IBD coefficient, that is inbreed coefficient. And if we know the genotypes of the individual and the allele frequencies. So from those three equations, we can get an estimate of Fz shown in these red equations. So the only information we need is the allele frequencies. If we know the allele frequencies, we know the genotype of the individual, we can get an estimate of the inbreed coefficient of this individual. But we can see the variation from a single locus estimate is very, very high. It is way out of, usually it is way out of the range between 0 and 1, or between -1 and 1, depending on your definition of inbreeding coefficient, whether it is a probability definition or a correlation definition. But the actual value of an inbreeding coefficient at a single locus is either 0 or 1. 0 when they are not identical by descent. 1 when they are identical by descent. And these occurs with probabilities 1 minus f and y and f respectively. For this particular example, if the allele frequency is 0.1 for the big A allele, the estimates of f is 11, 2.1, and minus 4.6. So depending on the observed genotypes. So it is very variable, as you can see. But the good thing is, although very variable, it is unbiased in expectation. At least, you know, when we know the allele frequencies accurately. And also the estimator is consistent. That is, with the increasing number of markers, the average inbreeding coefficient estimated from these markers will decrease consistently with the increase in the number of markers. But no matter how many markers we use, there is always a variance in the estimates because the markers are linked. We have a limited genome size, realistically. I think the most important thing affecting a marker relatedness or inbreeding coefficient estimation is not genome size or the number of markers. It is really the factor that allele frequencies, although assumed known, they are actually not known. They have to be estimated. There are quite a few problems associated with estimating allele frequencies from the same sample used to estimate relatedness and the inbreeding coefficient. I will illustrate this point later on. Now consider relatedness. For relatedness, we have to consider two individuals, X and Y. For each individual, we have two alleles and a locus. So in total, we have 4 alleles. Among the 4 alleles, there are 15 IVD states. If the paternal allele and the maternal allele are distinguished and are relevant. When this is not the case, when we do not know the origins of paternal and maternal alleles, or they are irrelevant, these 15 IVD coefficients, these 15 IVD states reduced to 9 condensed IVD states. So here they are, the 9 IVD states from S1 to S9. For S1, for example, the 2 alleles in individual X on the top line and the 2 alleles on individual Y on the bottom line, they are all identical by descent. So effectively, we have only 1 allele in this case. Is the probability of this state is denoted by delta 1. Similarly, we have S9, that is all 4 alleles are non-identical by descent. So we have 4 non-identical alleles. The probability of this configuration is denoted by delta 9. Because the individuals must have their 4 alleles must fall into one of these 9 IVD states. So these 9 coefficients must sum up to 1. And if we know these IVD coefficients, we can calculate the invariant coefficient of both individuals, individual X and individual Y. We can also calculate their coefficient of kinship and also their relatedness using this formula. Frequently in the literature, especially in the past, when we have few data, we assume invariant is absent. So delta 1 to delta 6 are always 0, and we only consider delta 7 to delta 9, and this considerably simplifies our estimation procedures. In this table, we summarize the IVD coefficients of some common relationships found in reality. For example, for parent offspring, we have only 1 IVD state possible, that is S8. Its occurrence is 1, probability is 1. So there is no variation in IVD between loci for the parent offspring relationship. So this is a very special relationship. Its ancestry coefficient is 1/4. For forceps whose parents are forceps, shown in this pedigree, we have all 9 IVD coefficients possible, and their invariant coefficient is 1/4, as I will show you later on, and their relatedness and their co-ancestry is 3/8. So this table shows the probabilities of each genotype pair of the individuals X and Y, given the allele frences and given the 9 IVD coefficients. For example, for the first pair, that is, they have the same, they share the same allele. They are all homozygote for the same allele. We have this coefficients available. And if you make a product between ABD coefficients and these coefficients, we get the probability of this occurrence of this genotype pair, which is here. So the first 6 terms involve inbreeding, while the last 3 terms is the case when there is no inbreeding. So they sum up, give this probability. Again, like inbreeding coefficient, these equations serve 2 purposes. One is for prediction of the pairs of genotypes, if we know the allele frences and ABD coefficients. And the other purpose is the other way around. That is, if we know the genotype of pairs, and we have the observed genotypes, and we know the allele frences, we can estimate from the data, from the genotype, from the allele frences to estimate ABD coefficients. And from the ABD coefficients, we can calculate relatedness and inbreeding coefficient. So again, they are based on markers. They are realized values for the particular markers that are used in estimating relatedness. But as I said before, if we do have a lot of markers taken at random from the whole genome, the average value should give a good estimate of the genomic or realized relatedness value. These estimates, again, are highly variable across loci, especially for full siblings, as I said before. But for parent offspring, it is always 0. There is no variation between loci. But the sampling variance can be considerably reduced if we have many markers available. That is why we need to, genotype many markers and use them in estimating relatedness, because it could reduce the variance a lot. And the quality of the R estimates is determined by many factors, including, for example, the real or actual relationship, and the marker information we have, how many markers we have, the allele frequency distributions of these markers, the missingness, and the error rate of these markers, and so on and so forth. And also by the related markers, related estimators we use. Different estimators use different methodologies, and they tend to yield different variance of the estimates. That is the way we see one estimator is better than the other, because it is more accurate. It has a lower variance of the estimation. And we need, anyway, we need many independent markers to produce a better estimate than a pedigree-based estimate. And as we said before, for marker-based estimation, we have implicitly a reference population there. Here, the reference is actually the allele frequencies which are used in calculating R. We usually assume these allele frequencies are available from the population, independent of the sample of individuals whose relatedness is being estimated. But in reality, this is not true. I will show you the problems with this later on. And whatever the reference, the mean really, if we use the current population as the reference, the mean relatedness would turn to be 0, and we would have a lot of pairs of individuals whose relatedness estimates would be negative. They are smaller than 0. This is because the mean is 0. Some are bigger than 0. Some are smaller than 0. Must be smaller than 0. So their mean is close to 0. That is because we are using the current population as the reference. If we use the ancestor population as the reference, ancestor quite a few generations in the past, then we would have a very low probability that a pair of individuals in the current sample has negative estimates of relatedness. So it's the reference problem. Yeah, so in terms of the negative relatedness estimates, it's better use the correlation definition rather than the probability definition, because some people, they tend to discard negative estimates or, truncate related estimates. When they are negative, they are taken as 0. I would say this is not suggested, because this would result in a in a bias in the estimates, and also these negative values do have meanings in the correlation definition of relatedness. They means they are less related than average. Over the years, there are many relatedness estimators developed and applied in practice. Here are just a few examples. For example, Witland developed a relatedness estimator to estimate delta 7, delta 8, and delta 9. And from there, we get an estimate of theta and r. And similarly, Queller-Goodnight, Lynch, and myself, Lynch and Witland, they all developed moment estimators of r and theta. And later on, Milligan developed a likelihood estimator to estimate these 3 IBD coefficients. And I developed also a likelihood estimator, but it could be used to estimate all 9 IBD coefficients. And from there, we can estimate invariant coefficient as well as relatedness. All these estimators have been implemented in my software called coancestry. And more recently, I developed a likelihood estimator to estimate delta 1 to delta 9, all 9 IBD coefficients, and jointly with allele frences. So we update allele frences, taking into account of the IBD coefficients, and use refined allele frences to re-estimate IBD coefficients. And the refined IBD coefficients is used to refine allele frences. So this is iterated over and over again until convergence. That is, we get better estimates of both relatedness and allele frences. I will show some more details of this later on. And also we have, yes?
Can you hear me?
Yes?
I have just one question. In your last method in EMBD9, in those estimates, zeros, I mean, there are no negative relatedness, right?
Yes. No, you're right. Yes, it's a likelihood method. There is no negative values there. But the good thing is that it updates allele frences iteratively. So in theory, the reference population is not the current sample being estimated for relatedness. Is some more remote ancestor population as allele frequency being estimated and being used as the reference. So we have a much smaller probability of getting negative estimates because we are using a remote ancestor population as reference. Does that answer your question?
Yes. Thank you very much.
Thank you. So there are also some other estimators developed more recently by many Kaiku in bioinformatics, implemented in the software called King. The good thing with this estimator is that it is much less affected by the small sample size. It works even you have just a few individuals sampled. You get almost unbiased estimates with small sample individuals. And also estimator developed by Konomos. It's about the relatedness estimation in unmixed population. It's implemented in a software called PC-relate. So here I show you, discuss with you some problems we have in estimating relatedness. Although the principle is simple, in reality, there are quite a few practical problems. The most important problem is in allele frequencies. You remember in the definition and in the development of the estimators, we always assume allele frequencies are available, and these allele frequencies are used as the reference for calculating relatedness. In reality, these allele frequencies are almost always available. We have to calculate allele frequencies from the same genotype data we have, which are used also for calculating relatedness. So we are getting the same information, both types of information, that is allele frequencies and the relatedness from the same sample. So there is a circularity here. We assume allele frequency available, but we actually calculate allele frequencies from the sample allele frequency, assuming individuals are inbred and are related. And from the estimate allele frequencies, which are assumed correct without errors, we calculate relatedness. So there is a circularity here, which leads to quite a few problems. I will clarify that later on. So we in the calculation, we assume the sample is large and the individuals are non-inbred and unrelated. Only in this, under this assumption, we can calculate allele frequencies by allele counting. For example, here, I'll show you a very simple example. We have 3 individuals sampled. At a single locus, we observed one big A allele and the rest are small A alleles. Because of this small sample size, the power of allele frequencies could be estimated with error. Usually, in the literature, we calculate the power of allele frequencies as the first formula. That is, we first estimate allele frequency as 1/6, and take a square to get the estimate of PA square. And this is actually assuming the sample size is large. in reality, you know, we have very small sample size here, only 3 individuals or 6 alleles. More appropriately, we should really use the formula that is derived under the assumption of sampling without replacement. This is the formula. So with the second sampling scheme, the estimate would be 0. It is smaller than the first value. And I showed this and in my paper published in Heredity, and that, you know, when you use the original estimate of powers of allele frences and in related estimates, you get a highly biased estimate when the sample size is small. Only when you use the second formula, that is sampling without replacement, you get unbiased estimates of powers of allele frences, and you get nearly unbiased estimate of relatedness. So here is a more complete description of the biasness of PA square. So if you use the first formula of sampling with replacement, that is, you're assuming the sample size is big, you get a biased estimate here. This is the expected value. It is always not smaller than the truth, that is PA square. But if you use sampling with replacement, you get an unbiased estimate, which is equivalent to the true value, that is PA square. Under the overestimation of PA square by sampling with replacement increases with a decrease in allele frequency PA, as shown in this graph. So the most bias occurs with rare alleles. That is one problem with a small sample size. It's relatively easy to correct for it. That is, we need to use the sampling with replacement that could correct for the bias. And the second problem is probably more difficult to solve. That is, although we assume a sample contains non-inbred and unrelated individuals, in reality, they usually contain related individuals or inbred individuals. And after all, that is exactly the reason we try to estimate relatedness. If we know they are all unrelated, then there is no reason to do that, to do the estimation. So here, let's see a very simple example. We have sampled 4 individuals. The first 2 individuals are full siblings, and the second 2 individuals are unrelated. And we have genotypes for a single locus with 2 alleles, small a and big A. If we assume all individuals are unrelated and non-inbred, we have allele counting to get the estimate of small a frequency, which is 5/8. But we know some of the small a alleles in the first 2 individuals are identical by descent. If we use this allele frequency estimate, that would be overestimate of small a allele frequencies, because small a, you know, some of them are identical by descent. This overestimation would lead to an underestimate of the individuals between full SIPs, that is the first 2 individuals, and an overestimate of unrelated individuals. So the best way to account for the relatedness between individuals is to account for their relatedness in the estimation of allele frequencies. So that is exactly what I did in my MEE paper published in year 2022. So basically, we know for full siblings, they have two pairs of genes identical by descent with the probability of a quarter. Only one pair of genes identical by descent with the probability of a half, and no genes identical by descent with the probability of a quarter. So in the first case, we have only two alleles that are independent. So we calculate the number of independent alleles here in the numerator, that is 2.7, 3.8, and 4.9 plus 1, because we have one allele here from in the non-inbred and unrelated individual. So we get an estimate of 0.571. This estimate should be closer to the truth. It gives a better estimate of the small allele frequency. This is just for illustration. In reality, we do not know whether the first two individuals are full siblings or not. So what we do in our paper is first, we assume, like previous approaches, all individuals are non-inbred and unrelated. And we, by allele counting, we get a rough estimate of allele frequencies as shown in the here, that is 0.625. And use this allele frequency to get a rough estimate of relatedness between individuals. And with the IBD coefficients estimates, we can update allele frequencies, use this formula shown here. And with this, we get a better refined estimate of allele frequencies. And with this better estimates of allele frequencies, we update relatedness and IBD coefficients again. So we do this iteratively over and over until convergence of both allele frequencies and relatedness estimates. So we can take into account all the relatedness in the estimate of allele frequencies, and we would get at last better estimates of relatedness. Or when I say better, I mean less biased and more precise. So the main problems in estimating relatedness, apart from the previous discussed issues, that is, we have to estimate allele frequencies from usually a small sample and usually a sample containing related individuals and inbred individuals. There is another problem with nowadays genomic data, that is, we could have a large genomic data which takes a long time to compute, even with a moment estimator, which is simple to calculate. For example, for the UK biobank data, we have 500,000 individuals and 59 million loci. So the data is 7,000 GB. Even they are in the most space-saving .bed format. So the most important thing is, we have 249 billion pairs of individuals for relatedness calculation. So even you can calculate and output 10,000 pairs of relatedness per second, you still need 145 days to do the calculation. One solution is to do parallel computation. In this way, you can use multiple CPUs, and also you can use all the memories in different CPUs So, so the storage of the huge data, as shown in the biobank data, could be solved. Also, the computing time could be reduced a lot. Another problem in related estimation is linkage and linkage disequilibrium. In developing the relatedness estimators, we always assume markers are independent. They are not linked, and they are in linkage equilibrium. In reality, because we are using genomic data, this is not true. All markers, they could be highly linked and in high linked disequilibrium. But I would say this is not a problem. And if we are interested in the point estimate of relatedness, not their confidence intervals, I wouldn't think linkage and linkage disequilibrium is important. It's not. And another issue is negative estimates. As mentioned before, this happens when we are using the current population as the reference. Actually, we are using the current sample as the reference. We do get a lot of estimates which are slightly negative, below zero. Again, I think as long as we interpret the results as, in terms of relatedness in the definition of correlation coefficient rather than probability, I don't think that is a problem. And I do not suggest truncating the negative values to zero. Another complication with related estimation is a subdivided population. Previously, we are talking about a single population. In reality, there could be subdivided populations. For example, two populations connected by, subdivided but connected by migration. So in this case, there are two approaches for estimating relatedness. One is a method I call the indirect method, developed by Anderson and Wang in year 2007, published in Genetics. So they use the population, the allele frences for the entire population, both A and B, under the differentiation between A and B, FST, to calculate relatedness between individuals within a population. But there is a direct method to do this. That is, we can take a sample from the population, either A or B, and estimate the allele frences, and use the estimate allele frences to estimate relatedness between individuals within that population. So this is the direct method. I showed in my paper, also published in Genetics, that even you have a small sample, so allele frences are estimated with poor quality, you can still get better results in terms of accuracy and biasness than the indirect method. If you are interested, you can read those papers to find out more. And another more complex situation is unmixed population. See here, I show an example. We have population A and B, and from A we have individual, and from B we have another female individual, and they interbreed to produce individual X and Y. Apparently, X and Y are hybrids. They are F1 individuals. Their relatedness, obviously they are full siblings, but if you use the allele frences from either A or B in calculating the relatedness between X and Y, you may well get a relatedness not 0.5 as expected. You have to take into account that they have different background, genetic background. They have mixed background. So you have to calculate the individual specific allele frequencies of both A and both X and Y, and use those individual specific allele frequencies to calculate relatedness. That is the principle. That is the idea used in these three publications in the three software, RIP, Relate Admix, and PC Relate. They can take into account the admixture of the individuals and their ancestries to calculate relatedness. So I guess after this discussion, we can have some conclusions or take-home messages. One is that there are a lot of marker-based methods available, and they are used in estimating relatedness in different applications. They are based on different assumptions, and they have advantages and disadvantages. We cannot say one estimate is always better than the others. We need to find out, in the particular situation, which one is better to apply. And that is why in some software, they built in many estimators for the user to investigate of their particular data set and to make a decision and to make a comparison. And another conclusion we can reach is that the precision, accuracy of a marker estimator increases with the use of more marker data, more markers. So when we have a lot of marker data, we can get more accurate estimate of the genomic or realized or actual relatedness than a pedigree-based estimate. Roughly, if we have, say, for example, 10,000 markers, they are of high quality, of course, no missing data, all those ideal situations, we can get a better estimate of relatedness than pedigree-based estimate. And also with the use of genomic data, we can get detailed estimates of IBD coefficient. We can get estimate of all 9 IBD coefficients. So we can take into account our inbreeding coefficient, and we do not have to assume inbreeding is absent, which is not always true. And relatedness is defined and measured relative to reference population. I emphasize this point over and over again because people tend to forget this. Relative relatedness is really a relative thing. It is always relative to a reference, no matter you are aware of it or not. If we change the reference, we change the relatedness estimates. And when we get some estimates of relatedness, they should be interpreted with this reference population. And many of the issues with relatedness estimators are actually due to allele frequency estimation. Although we assume allele frequencies are available without errors in the estimation, they are actually not true. We have to estimate allele frequencies from the single sample of individuals from which we are estimating relatedness. So there is circularity there. And also, most often, the sample available for our estimation is small, and they contain inbred and related individuals. They also contain duplicated samples, and there could be also population structure and mixture. All those complexities could be possible. So when these complexities are possible, we need to take certain measures to reduce the problems before we can conduct a good estimate of our relatedness. Louis pressed me for talking about missing data, but because of the time, I didn't mention this. But I would say missing data is important for one reason, simple reason, that is, because when we estimate relatedness, we must use data from both individuals. If data is missing for one individual, then the data is useless for that locus. So if the data missing rate is 50%, so then if the missing rate with the data missing is random across individuals, we have only 25% of data available for estimating relatedness for a particular pair of individuals. So missing data has a large impact on the data quantity available for relatedness estimation. Yeah, there are some issues with computer efficiency with larger samples in relatedness estimation, but I guess this is the only relevant for human data set. Currently, it's not a problem with conservation, for example, because samples are always not too big, but actually too small. But we have to bear this in mind that computation could be a problem when you have very large samples, because we are talking about computational efficiency of pairs of individuals, not a single individual. So it increases with the square of n, not just n. I guess that's all. I would be happy to take questions from you, and after that, I guess Louis and Natasha will show you more interesting stuff of their analysis of some empirical data sets. Thank you.
Thank you, Jinliang, for having time to come to talk with so much experience you have on this issue. And anyone has any question in the room here or online?
Thanks a lot. That was really interesting. I've got a question regarding, because in the workshop we came up that it's actually quite, due to lab processes and also bioinformatics, it's really difficult sometimes to call SNPs with certainty heterozygous. And my question is how this would affect your estimates in relatedness if you can't be sure about heterozygous individuals at a low time.
Yeah, so effectively, it's when you have difficulties calling heterozygotes, so heterozygote can be called as a homozygote. It is effectively similar to the situation of macrosatellites, that is, allelic dropout. I guess it's mentioned before in this workshop. So basically, we get more homozygotes than the truth. In this case, if every marker is acting like this, we could get biased estimates of relatedness. But if you have difficulties of calling heterozygotes with just a few or a small fraction of the SNPs, most other loci are called correctly, then relatedness should not be biased too much. It should not be affected too much. Its relatedness estimation is different from genealogical relationship inference, like full sibling inference or parentage inference. With those relationship inference, genotyping errors, allelic dropouts, these factors could affect a lot if you are not aware, if you do not aware of the problem and take appropriate measures to account for the problems. There could be serious problems because a single locus error could cause a serious error with your inference. But for relatedness estimation, we get estimates from each locus, and we make an average across loci. So if there are errors with just a few loci, the average should not be affected much. Does that answer your question?
Perfect. Thanks a lot. That was the answer I hoped.
Thank you.
We have another question here.
Hi, Jinliang. Bill Sherwin here. Thanks very much for that. That was great. I didn't quite catch what you said about linkage disequilibrium. I'm wondering if you might comment on those 10,000 markers. Are you saying it's not relevant whether they're statistically independent of one another?
Okay. Yes. With 10,000 markers or even more, definitely there is linkage disequilibrium there, and but its impact on relatedness estimation really depends on your focus. I guess for most people and for most applications, what we are interested is in the point estimate of relatedness. See if we have individual A and B, we just want to know on average how related these two individuals are, and see is it 0.1 or 0.5, and very clearly people are interested in knowing how reliable or the confidence interval of these point estimates. If we are really interested in the confidence intervals around these values, then linkage disequilibrium should be important, because linkage disequilibrium, these loci are correlated. They give correlated information. So if we assume they are unlinked in linkage equilibrium, then we would get a confidence interval that is too conservative. But as I said before, as I argued before, most people are actually interested in the point estimate, not in the confidence intervals. Yeah.
Okay, thank you.
Thank you.